Many sporadic cancers are developed when the cell's DNA is changed or mutated. Some carcinogens act directly on DNA, causing the damage and a subsequent mutation, which results in tumor growth. The ability of carcinogen to cause cancer depends on many factors, including the amount of exposure, the length of the exposure, and the genetic makeup of the individual. Carcinogens include chemicals such as asbestos that lead to lung cancer, medical radiations used in diagnostic tests, viruses such as hepatitis C causing liver cancer, and lifestyle factors. The most prevalent carcinogen in our natural environment is ultraviolet radiation in sunlight. The skin cancer melanoma caused by prolonged exposure to ultraviolet radiation has the greatest number of somatic mutation per tumor. Recent study identified the solar signature patterns of mutation in DNA sequence of skin cancer cells, mainly cytosine and thymin. The two primary classes of mutagenic DNA lesions caused by ultraviolet radiation are cysteine cyclobutene pyrimidine dimer, or CPDs, and 6,4 photoproducts. CPDs, however, are the most frequent UV-induced DNA lesions. In the case of melanoma, the UV-induced mutations are not evenly distributed across the genome, which indicate that the genomic and epigenomic features associated with these mutation sites are likely to regulate mutagenesis processes. Therefore, features regulating localized genomic carcinogen susceptibility play a significant role in the development of a tumor by determining the probability for mutations. These features include deficiency in repair or replication, decreased nucleotide occurring at the end of S phase, and transcriptional activities such as transcription copper repair. It is generally accepted amongst most studies that insufficient DNA processing and repair mechanisms are the major contributing features resulting in mutagenesis. However, some studies also indicate that chromatin-bound factors can influence the susceptibility to obtain UV-induced DNA damage in the first place. The current study presents the first genome-wide mapping of DNA lesions induced by UV radiation in human genome. The study has discovered considerable variation in distribution of UV-induced lesions through a comparative analysis of carcinogenesis rate in lamina-associated heterochromatin and euchromatin. IMR90 primary fibroblast cells were exposed to ultraviolet radiation followed by immediate cell lysis. The DNAs were then purified for immunoprecipitation processing. Following photolase repair, genome sequencing was performed. Chromatin states were also assessed as part of the epigenetic analysis, with chromatin features including RNA abundance, DNA methylation, and 16 histone modification. These were followed by lamine assessment, 3D genome modeling, nucleotide excision repair rate measurement, and melanoma analysis using whole genome sequencing data from 25 melanoma tumor samples. The results demonstrate a negative correlation between UV lesion abundance and detection of the repair genome void, which verify repair deficiency in heterochromatin. While lamina-associated heterochromatin is highly susceptible to the carcinogen, the euchromatin is relatively protected from the ultraviolet radiation. Many cancer driver genes have an intrinsic increase in carcinogen susceptibility. The high mutation rate observed in a heterochromatin is not only due to decrease in repair efficiency, but also to an increase in the accumulation of DNA damage in these regions. This can explain the significant contributions that carcinogen susceptibility makes to the mutational variation in cancer. This diverse mutational spectrum is largely predetermined by inherent susceptibility to the carcinogen, while the degree of susceptibility itself is controlled and regulated by genome architecture. This figure shows the correlation of abundance for indicated features to principal component between the euchromatin and heterochromatin. It specifically shows the tumor suppressor are significantly more susceptible and highlights the major contribution of lamina association and chromosomal position to cancer development. Overall, the results from this study demonstrate that the epigenome architecture acts as the most important barrier to cancer development by regulating susceptibility to carcinogen in the non-malignant cells. Future studies may uncover therapeutic strategies to decrease carcinogen susceptibility, therefore prevent or suspend the initiation of tumor development.